viewers, and welcome back to another episode of Health Watch. For today's episode, we will be having a special report on the malaria virus, a dangerous virus caused by a microscopic parasite that has affected millions of persons worldwide. Our team of researchers has the latest updates, statistics, and research on this deadly disease. I now hand over to Miss Hammond, who will tell us more about the parasite that causes malaria.
Thank you, Miss White. Plasmodium parasites are the cause of malaria. Two of the five Plasmodium parasite types that cause malaria in humans are P. falciparum and P. vivax, which are the most dangerous. The most common malaria parasite in Africa is P. falciparum, which is also the deadliest. In the majority of the nation outside of sub-Saharan Africa, P. vivax is the predominant malaria parasite. P. malariae, P. ovale, and P. knowlesi are the other malaria species that can infect humans. This parasite is a major global concern because malaria continues to affect millions each year, especially children under 5. The disease can cause severe illness and often lead to death. Understanding how malaria spreads and behaves is crucial for developing better prevention and treatment strategies. According to the global report, malaria remains a significant health challenge. In 2023, an estimated 263 million cases were recorded worldwide, leading to 597,000 deaths, a slight improvement from the 600,000 deaths in 2022. The World Health Organization African region remains the most affected, accounting for 94% of all cases and 95% of all deaths. Children under 5 make up about 76% of malaria-related deaths in the region.
Okay, thank you, Ms. Hammond. So, artificial intelligence was integrated throughout this research, but I now pose the question, what is artificial intelligence? Well, according to TechTarget, artificial intelligence is the simulation of human intelligent processes by machines, especially computer systems, including learning, reasoning, and self-correction. So, what are some types of AI tools? Well, we have three examples here: deep learning models, generative model, and the large language model, such as ChatGPT and Gemini. For our research, DeepSeek was the selected tool as it provided advanced reasoning and accuracy. In addition to that, it is reliable, inexpensive, and was readily available. The malaria virus was selected from the provided list of microbes and their diseases and was selected as at the initial stage of research. It provided a wide range of accurate and current information relating to Jamaica. AI was also used to pinpoint additional scholarly sources throughout the research in order to provide the other necessary information. So we also found some statistical information relating to the malaria virus. According to Mayo Clinic Staff 2025, malaria is caused by the protozoa of the genus Plasmodium, of which these four are pathogenic in humans: F. falciparum, P. vivax, P. ovale, and P. malariae, and is transferred to humans by the bite of a female mosquito from the genus Anecia. Each year, there are approximately 2 million to 5 million new cases around the world, with the disease being linked to 1 million to 2.5 million deaths annually, and that is according to the World Health Organization 2024. On a global scale, majority of infections stem from the P. falciparum and P. vivax, with the P. falciparum being the most widespread parasite in West and Central Africa, having 100%, as well as high transmission in East and Central Africa, also 100%, and Southeast Asia, approximately 50%, whilst P. vivax is the dominant in the Americas, 70%. World Health Organization 2024. Locally, from 2006 to 2009, there were 406 confirmed cases, where 90%, or 361 cases, were from residents living in Kingston and were as a result of the P. falciparum, according to the Public Health Ontario 2025. I now hand over to Miss Burton, who will take us through the mode of transmission.
Thank you, Miss White. Malaria is transmitted normally via the bite of an infected mosquito, and in this case, mosquitoes act as vectors. And vectors are living organisms that can transmit infectious pathogens between humans or from animals to humans. The parasites at this part of their life cycle is known as sporatocytes. They are introduced into the host's blood when the mosquito takes a blood meal. From there, the sporatocytes travel to the liver, they reproduce, and this process may take several weeks to accomplish. Then they finally re enter the bloodstream, and at this point, the patient will begin to experience symptoms. There are other less known modes of transmission of malaria, such as congenital malaria. This is from mother to infant during pregnancy or childbirth, transfusion malaria through contaminated blood transfusions, and needle stick injuries or contaminated needles. Now, passing it on to you, Mr. Melvin. You guys?
So good day. Thank you, Miss Burton. My name is Omark Melville, and today we will be taking a deep dive into the complex life cycle of the Plasmodium parasite. So let's begin. Our journey begins with the sporozoite. This is usually injected by a mosquito, and this is the primary infective form. It is a slender, spindle-shaped cell, 10 to 15 nanometers long. Its surface is covered with proteins like the circumsporozoite protein, or CSP for short, that are essential for gliding motility and for recognizing and invading liver hepatocytes. This form is built for speed, stealth, and targeted invasion of these hepatocytes. So, once inside the liver cell, the sporozoite transforms, initiating something called the exoerythrocytic schizogony. In this phase, it loses its slender shape, becoming a large, metabolically active trophozoite that undergoes multiple rounds of nuclear division without cytokinesis. This creates a massive multinucleated liver schizont. This schizont is a true replication factory, eventually containing thousands of the next generation. So when mature, the liver schizont ruptures, releasing thousands of merozoites. These are erythrocytic invading forms. They are small, only about 1.5 nanometers in size, and are ovoid or pear-shaped. Their most critical morphological feature is the apical complex. This is a specialized set of organelles, and upon contact with a red blood cell, the apical complex discharges its contents, creating a moving junction that actively pulls the merozoite into the cell, thus forming a protective parasitophorous vacuole. So now I'll be going more in detail on the blood stage morphology, where I just talked about the liver schizons and more. So let's begin. Inside the red blood cells, the merozoites swiftly develops into the ring stage trophozoite. This classic signet ring morphology consists of a thin circular loop of cytoplasm and a prominent red staining chromatin dot, which is the nucleus. This stage is primarily for nutrient uptake and establishing the infection with a new host cell. As it matures, the ring becomes a larger trophozoite. Its morphology becomes increasingly amoeboid with pseudopodia-like extensions. In this state, this is the main feeding stage of its state like this. So then the parasites digest up to 80% of the host cell hemoglobin in an acidic food vacuole. The toxic heme is then crystallized in an insoluble inert pigment called hemosin or malaria pigment. The accumulation of this brown to black crystalline pigment is a key diagnostic feature under the microscope. Then the mature trophozoite undergoes erythrocytic schizogony. The nucleus divides repeatedly, leading to this schizont stage. An early schizont has multiple chromatin dots. A mature schizont is really a very spectacular sight to behold. It nearly fills the red blood cell with a cluster of 8 to 32 merozoites, surrounded by a coarse, dark clumps of hemosin pigment. The rupture of the schizont is what causes the symptoms of fever and chills. So finally, some merozoites commit to transmission by developing into gametocytes. This is the sexual stage, and they are larger and often fill the red blood cells in Plasmodium falciparum. The macrogametocyte, the female counterpart, is typically a crescent or banana shape with a compact nucleus, while the microgametocyte is the male counterpart. This is more sausage shape with a diffused nucleus. The distinct crescent morphology is a critical diagnostic differentiator. So we'll now be moving on to reproduction in the mosquito host. So, when a mosquito ingests these gametocytes from a blood meal, the warm, oxygen-rich environment in the midgut triggers gametogenesis. The microgamete undergoes a rapid process of exflagellation where in just 10 to 15 minutes, its nucleus divides to produce 8 flagellated motile microgametes that whip free from the residual body. The macrogamete matures into a single larger, no, the macrogametocyte, I should say, matures into a single larger macrogamete. Fertilization of the macrogamete by the macrogamete forms a diploid zygote. This zygote, within 18 to 24 hours, transforms into the motile vermiform worm-like okinete. The okinete elongated morphology is essential for its function. It uses proteases to penetrate the mosquito's midgut lining and escape the blood meal. On the outer wall of the midgut, the okinete transforms into a round oocyst. This structure is stationary and protected by a resilient capsule. Inside, another phase of asexual replication called sporogeny occurs. The nucleus begins to divide repeatedly, and the cytoplasm coalesces around these nuclei, producing thousands of slender haploid sporozoites. After 10 to 14 days, the mature oocyst ruptures, releasing the sporozoite into the mosquito's body cavity. These sporozoites then actively invade the salivary glands of this mosquito, ready to be injected into a new human host, thus completing the entire cycle of this parasite. So, in summary, the malaria parasite is a master of morphological transformation. Each stage, from the invasive sporozoite and merozoite through the diagnostically critical ring trophozoite, schizont, and crescent-shaped gametocyte to the motile okinete and replicative oocyst is a precisely adapted tool for survival in each stage and also replication and transmission. This intricate dance of form and function is what we are up against. So thank you and be safe. Now I'll pass on to the next presenter.
Thank you, Mr. Melville. Today I'll be giving a quick overview of the main tests used to identify Plasmodium, the parasite that causes malaria. There are four major diagnostic methods that we use. First, microscopy. This is done using thick and thin blood smears stained with Gemza, and it is considered the gold standard. Gemza is a microscopic stain used in hematology. The thick smear concentrates the blood so even low parasite levels can be detected, where the thin smear spreads out the cells to help identify the specific Plasmodium species. This is referred to as the morphological examination. Next, we have rapid diagnostic tests. We call these RTDs. These detect malaria antigens. This test resembles a pregnancy test with more than one line indicating a positive test result. However, their accuracy can be affected by storage conditions, species differences, and the quality of the kits and results should be confirmed with a blood smear. A more advanced method is PCR, or polymerase chain reaction. PCR detects the parasite's DNA, distinguishing between all five species. It can identify species accurately and pick up very low-level infections that other tests may miss. PCR diagnosis of Plasmodium begins with extracting parasite DNA from a blood sample and then amplifying it using the polymerase chain reaction so even tiny amounts become detectable. Then we identify the exact Plasmodium species present. Finally, there's the quantitative Buffy coat or QBC test. This method uses a special fluorescent dye and a centrifuge to concentrate parasites in a specific layer of the blood. QBC is faster than standard microscopy, but it can be more expensive and doesn't always clearly distinguish between Plasmodium species. So each method plays an important role. Microscopy remains the standard because it is affordable and provides detailed information. RTDs are quick and practical for rapid screening. PCR is the most accurate for confirming infections and identifying species, and QBC offers fast detection where the equipment is available. Now, let's shift to the temperature requirements of Plasmodium. Temperature plays a major role in determining where malaria can occur and how quickly it spreads. Inside the mosquito, the parasite develops in warm conditions, generally between 25 and 30 degrees Celsius. At these temperatures, the parasite matures faster, which shortens the time before the mosquito can transmit the infection. Each plasmodium requires a minimum temperature to complete its life cycle. If temperatures drop too low, typically between 18 to 20 degrees Celsius, for P. falciparum, development shows or stops completely, preventing transmission. On the other hand, extremely high temperatures above 34 to 36 degrees Celsius can reduce mosquito survival, which also limits the spread of malaria. Different Plasmodium species have slightly different temperature needs. For example, P. vivax can tolerate cooler conditions and may develop at temperatures as low as 15 degrees Celsius, which is why it can occur in more temperate regions. P. falciparum, however, is more sensitive to cold and thrives mainly in warmer tropical climates. Overall temperature influences both the mosquito's activity and the parasite's development, which is why malaria is concentrated in regions where the climate consistency supports both.